I am thrilled to be moderating our last session for today. One of the things that we've heard in almost every presentation and repeatedly throughout the day is the importance of translation, and that is the third of CIMBID's pillars, and that's what we're going to be talking about today in our final session. So without further ado, I'm going to ask Stella Kang to come and tell us about cost-effective approaches for clinical translation of AI-based imaging tools. Thank you.
All right. Figure out advancement here.
Just on
the green one, right?
Just here on the screen.
Oh, the screen. Okay. Awesome. Thank you. Very nice to see you all. Good afternoon. I'm Stella Kang. I am a body imager. That's my clinical background. I was brought here at the very end of 2024 to be vice chair for clinical research in the Department of Radiology and to start a new center in the department. I'll talk about a little bit and how it will synergize with CIMBID. So we are really at a frontier, and it's very exciting to hear about CIMBID and all that it is really poised to contribute to AI, certainly to our medical center, and with new programs and development and opportunities for many people, but also what it's going to contribute to diagnostic imaging and to AI for healthcare. And while we're very excited to see the progress in AI and all that it's doing, diagnosing cancer, flagging critical findings, integrating clinical workflow in ways that sounded like science fiction even 10 years ago, we're also in a place, at a medical center, where we're very well poised to look at some very hard questions of the technology next. So after we know that it can find the disease, we have to answer questions like, is it really improving the care for patients? Is it really delivering the impact that we want to see in patients? So what we're interested to do in our department is to develop the AI algorithms and to test them and to look at their performance, but then to look in the real world setting and to look at their comparative effectiveness and their impact on their clinical outcomes, right? And hopefully we can see with the two centers here, the Center for Advanced Diagnostic Research, that among these questions that we'll be poised to ask are the tools that are coming out of CIMBID, are the tools that we're all working to bring to patient care, really making the impact that we would like to see. And it really is a sort of a journey for the tool. Of course, at the beginning, we have to make sure that the tools are useful to clinicians, right? It has to be seen as adoptable, have face validity, and if it really doesn't pass that first initial step, it doesn't have much hope of delivering the impact that we'd like to see. And then along with all those other important tests, diagnostic accuracy, right, smarter decisions overall, we of course want to see that the care is expedited. Are we reducing unnecessary interventions? Are we just introducing new ones on the flip side, right? And are we reducing the time to the critical treatments that are necessary to treat the correct disease once it's diagnosed? And eventually we want to come to a place where we find that the patient's ultimate endpoints, their survival, right, or cause-specific mortality, those are also improved. And that's where comparative effectiveness research comes in, and hand is the cost-effectiveness research. It was really born out of an interest in assessing health technology in such a way that we're directly comparing new and often more expensive technologies to intervene on a disease or tests that are meant to diagnose the disease against the current standard, right? Sometimes the current standard is we don't have a test. We just have what we use as a heuristic or our best clinical guess, right, or a simple laboratory test, and then we go with that. And sometimes there is a pretty well-developed test, and what we're interested to know is if that test can perform better, or if that treatment combined with the test can yield faster and more effective therapy. And typically, the outcomes will focus on gains in health, right? So life expectancy, survival, or reduced mortality that's cause-specific. And we might also look at intermediate outcomes, such as differences in diagnostic accuracy, or even patient-reported outcomes, as well as other relevant outcomes to the healthcare system, for example, cumulative costs. So here's a typical patient journey right? You arrive, you have some sort of question in your mind, for the clinician's perspective, whether this patient has one of many diseases on the differential diagnosis. We have some clinical decision rule, classically. Do they have this abnormal lab result? Do they have this combination of physical exam findings plus the abnormal lab result? And then a number of different things happen. They have their clinical presentation. The clinician has to make some sort of decisions about what test to order next. Then there's a change in the patient's status over time. There's the treatment decisions based on the synthesis of all those things that have happened so far. And then the patient's disease response, right, to all of those decisions and the treatments that have been administered. And ultimately, they'll arrive at some sort of a clinical endpoint. So in basic terms, we start to overlay some of the measures that we're interested in. That can be markers of whether a new test or a new clinical decision rule, here we're talking about adding, for example, AI risk analysis, might improve some of these metrics that are placed along the pathway. And these are classic intermediate outcomes. So diagnostic certainty, right? Are the clinicians finding the tool so believable, adoptable, useful, so that they're more certain of what the diagnosis might be, and therefore are much more directed towards that pathway, right, from their initial very broad differential diagnosis? Are our invasive testing decisions changing? Are we ordering less of the classically ordered invasive tests? Or are we seeing a disbursement where we have different versions of testing, perhaps positively towards the less invasive, less costly options, or even just follow-up? And are the treatment decisions then correct and directed towards the diseases that are being correctly diagnosed? And then, are we seeing improvements in the patient's safety in their course of care, the efficiency of that care, and also the cumulative costs of that episode of care? So all of these things can be measured along that patient's particular journey. And eventually, we want to also have, of course, some clinical endpoint that's relevant. And ideally, we want to look at something that's a surrogate. If it's not something that's so critical so that we expect high and rapid mortality, we can still look at surrogate endpoints that are important markers of health, right? So sometimes this comes in the way of detecting a cancer in an early stage, or reducing some sort of invasive therapy that may not be so helpful in the way of prolonging life expectancy. So I'll just take one illustrative conundrum that we probably encounter most often in imaging, and that's the incidentaloma. So incidental findings are lesions that are found on diagnostic medical imaging that have nothing to do with the original clinical reason for the scan. So say, for example, a patient has abdominal pain, they come to the emergency room, and they get a CT scan right away. These days, that's true. They don't even do the physical exam. They're often sent right to the scanner. And typically, we find a lung nodule, right? We find some gallstones. We find a number of other things. They're highly prevalent, typically found, I would say, in the journals between 20 to 30% of the time on scans. And in some subgroups, they're found in virtually everyone. Okay, so we have thyroid, lung, every organ can have them. And so these are a problem because we have to find them and report them and synthesize what it all means to the patient, if anything at all. So eye test, especially for those of you in the back. This was a patient who came in with dizziness and weakness and got a CTA, a CT angiogram of the neck. Anyone see anything? Yeah, our neuroradiologists will spot it in a second. This is extremely common. Okay, so in that left thyroid lobe, there's a little nodule. I don't know if this will work here, right there, that little dot. Okay, so our very astute radiologist takes a closer look, and indeed there's something there. It's a little less than a centimeter. And so typically when I poll a room of radiologists, I ask who would report this, who would even notice it, and who would report it. So about, I would say typically half would find it and honestly admit if they would report it or not. But this is why it's a conundrum, because in this case, the radiologist did recommend an ultrasound to characterize the nodule, and it has for us radiologists, these telltale microcalcifications, and it has a lot of flow in it. And so this is a classic this is a papillary thyroid carcinoma. And it's less than a centimeter, right? So it's kind of on that border of what we would even find reportable when we're talking about what our current practice guidelines say. So we typically rely on evidence-based, but pretty simple guidelines to date, because that's what we can do, right? When we're reading a high volume, we're coming across many of these, we gotta go on what the typical course of a thyroid nodule will be. So it's fair to say that half might report it, half might not notice it. And the question is, if the AI can find more of them, does that solve the problem? Or if the AI can diagnose more clearly and accurately what the probability of malignancy is, does that solve the problem? And in both cases, probably the answer is no, right? Because what we know from screening programs that have been attempted outside the US, thyroid cancer screening does not improve survival, cause-specific survival for thyroid cancer. But there is a difference, right? Some of these cancers turn out to be pretty aggressive. Most of them turn out to be, rather indolent, but they can spread locally. So there is a heterogeneous course that perhaps AI could find a role for and integrate care in a more effective fashion. So this is where our comparative effectiveness, our prospective studies become so important, because we want to know not just the diagnostic certainty or the probability of malignancy, but whether the smarter choices are coming out of use of the tool, whether the treatment decisions are yielding more effectiveness in the way of patient care, and of course, overall, keeping it within cost. And just a brief note is that we find over and over in technology assessment that technology can be more expensive, but as long as it is providing smarter care, right, delivering that effectiveness that you bring with the cost, it can still absolutely be cost effective. And so I'm excited for CIMBID. I'm excited for the pursuit at our medical center for these technologies to deliver AI that's going to be not just promising, but with proven impact, and to find the evidence to make it trustworthy. Thank you.
Any questions? Yes. I got to wrangle the speakers too. Yes.
So the time thing.
Hello, yeah. So I had a question.
Who's the mic?
You were talking, so I'm sure, yeah, sorry. I'm sure it's usual, when there's a new technique, doctors are formed as we teach them how to use it and make new decisions. But with AI, if the technique is supposed to do what they are already measuring, but much better, see it like the measurement of the length of the tumor can be only underestimated if you mess up the search for the longest step. Whereas, so if we all use that technique, all the tumors are going to grow by, 3% tomorrow, right? Just because it's more accurate. So are there like some ways to account for that? Because it's not completely a new technique, it's doing what they do better. Or if we switch to volume of the kidney instead of length, then percentage are not the same because it's 3D. Like doubling the length is much more dramatic than doubling the volumes and those kind of stuff.
Yeah, I see like 7 questions wrapped up in your
sorry.
No, don't be sorry. It's all important stuff. I mean, I think that, the illustration of the adult polycystic kidneys was interesting because, you have the volume of the cysts, they're growing. They're not necessarily all replacing kidney parenchyma. They stick out, so you don't care as much how much they're sticking out, right? There's lots of metrics that are needing to be joined together, right, to find the useful part of the kidney parenchyma that's still functioning. That's one of our most important questions in those patients. So that's just one example. And the same is true for oncology, right? This is why we have very complex ways of measuring tumor growth and making sure that they meet some threshold. So in your question, I see that we have metrics that have to make sense together. Sometimes it's not just one number. It's a combination of features that make sense anatomically and physiologically. And I also see that there's thresholds. Sometimes clinicians all are living on a different place in terms of threshold and the ROC curve, right? And so with AI, we have opportunities to calibrate, We have opportunities to make sure that we're performing in a range that makes sense to maximize the clinical benefit, right? And all of those things can be followed and measured, I think, alongside the technology development. And so hopefully that's a brief answer, but there's a whole seminar that could be discussed for this.
Thanks.
I did not notice the thyroid nodule. I just thrown it. I hope I'm not the only one in the room who didn't. But I thought the framing in that talk of, does this make a difference? Is this changing patient care? I think that's important for us as we sort of think about what the goal of translation is. Next up, I would like to welcome Bethany Percha, Chief Data and Analytics Officer at NYP. Thanks.
Great, thank you so much for having me. I have to offer a little bit of love to Despina, of course, like many of the previous speakers. I have been here for a little over a year. Despina was one of the first people I met when I came to the health system, and I was just really inspired by her energy and vision for the center. Really excited for all of the work that's going to happen here. I basically am going to spend my talk on this slide mostly. I have a few auxiliary slides. But I'm really here to offer the perspective of the health system in all of this. So I don't know how many of you are Columbia, Cornell, NYP, but if you're not any of those things and you haven't gathered by now, we have a tripartite structure. So we have one health system. I'm employed by New York Presbyterian, the health system, and then we have two Ivy League great medical schools that are both affiliated with us. And that structure has some advantages and some disadvantages. One of my colleagues who's on the Data Shared Services Oversight Board referred to the two medical schools as ships both orbiting the Death Star. The health system being the Death Star. I thought that was appropriate. But yeah, a lot of the presentations earlier have referred to data sets and clinical translation of work that really intimately involves the health system. So the Data Shared Services effort is why I was brought here. Around 2019, I think, 2020, predates me, the three institutions got together and decided to implement a shared EHR. And so they implemented Epic across the three institutions. And they put a team together called Clinical IT Shared Services, which runs the EHR, everything from reporting to application development, et cetera, within the EHR. And they developed a whole structure around this for how it would be funded, how the different employees would work. They could work at different institutions. And that led to a lot of success. They were actually able to deprecate, I think, 23 different EHRs throughout the health system and implement a single one. And that team has been centralized and has a governance structure after that that's been working really well. And so the team that started that shared service wanted to extend that to other areas. And the data shared service is the next one on the list. So as the health system and as our school partners interested in translational research, how do we put a set of structures and infrastructure and services in place that allow us to do translational research more effectively, that allow us to operate more efficiently, and that allow us to avoid duplicating effort across three different places, right? We have, for example, operating analytics teams or operational analytics teams at both schools and at NYP. A lot of the work that they do is overlapping or redundant in some way today, but we are seeking to kind of consolidate that, to get people working together, to create a common governance mechanism for our data so that we can do things more efficiently and ultimately help our patients and promote translational research. So there are 4 parts to the data shared service. The first is our platform team. So we are building a new platform to provide data to both schools and other teams at NYP. That platform, today we have a legacy SQL Server architecture to do this. We're going to be building a new platform in the cloud. It'll use modern cloud technologies and be scalable, and it will offer all of our clinical and operational data in a common place through a common governance mechanism that is overseen by all 3 institutions. So that's a tall order. It'll take us a long time to get there, but we did engage in a tripartite RFP last summer. We chose our technologies for the data platform, and we're actively in contracting right now with funding from the health system to build it. The second thing we did is we restructured all of the central NYP operating and reporting teams that were either engaged in like work for Epic reporting or Tableau reporting. There was a kind of a mishmash of teams there. We restructured that around these strategic pillars. You can see 7 there. And what that allows us to do is for our central shared service team to partner more effectively with operational teams elsewhere in the health system and with our school partners to engage in really targeted analytics geared towards specific areas like hospital operations. Otherwise, we would have requests coming in for various reports. They'd all be handled on an ad hoc basis, and it was much less efficient. We think that with this structure, we'll create a machine that will allow us to communicate with our school teams, with our faculty practices, et cetera, a lot more efficiently. A third thing that we are going to be providing as part of the shared service is really support around translational research. One of the common complaints I hear from my faculty colleagues is we do all this great research, we get it right to the clinical deployment, and then it becomes impossible because every single project is being done on a bespoke basis. Right? You have to have a set of negotiations with various committees. There's no kind of common deployment architecture, especially with data science projects. So it's just a lot slower than it needs to be. And so what we'll be providing as part of the shared service is our clinical and research informatics team. Karthik is here, he leads that. And really that's about taking all these bits and pieces, new patient cohorts, insights from natural language processing, various things that have been done at the schools in research that augment our data model and help bring them and centralize them within the platform. So one example is George pointed out OMOP, right? OMOP is a common data model. Today we have 2 OMOPs. We have one at Cornell and we have one at Columbia. Really, the logic for that should be centralized in a single place, and those teams are actively working together to help build that into a common architecture within the new data platform. We're also investing in certain strategic translational data science areas. The health system has made substantial investments in cardiac AI, so we support teams both at Cornell and Columbia, and that technology has really spurred innovation, and it's actually produced some commercializable results as well. So we'll continue to do that for strategic areas. I'm proud to say that we are supporting both Stella and Despina's centers with staff and time and resources because we believe this is another really important strategic area for the health system. And then finally, we have a team dedicated entirely to evaluation. I chair our newly founded tripartite AI governance committee, so 15 people across all 3 institutions. You can imagine how easy that was to get off the ground. And we have a team within the shared service whose job it is to basically design evaluations for new technologies and make sure that we're testing them accurately and keeping our patients safe. And then finally, we're offering a data operations and outreach function. This is sort of a wraparound for a lot of stuff we already have, like TRACK, our tripartite data access requests. Our registries and regulatory team, which manually curates our clinical registries, lives here. We have over 50 registries that we support at the health system level. Project management, and then we're really investing in kind of a common look and feel to all of our data assets, and a data literacy program as part of this service. So really, that's the bulk of what I wanted to tell you. This is ongoing. I have a team of 200 people, if you can believe that, mostly based at NYP, but also with partnerships with a lot of school teams. We have an open door policy to our team meetings. We welcome school researchers and others who are engaged in operational analytics or clinical research to attend our retreats and everything that we're doing as part of the shared service. We are engaged in legal contracting right now for 3 of our platform technologies. We're actively implementing a data governance technology called Elation, and there's a ton of momentum and just support from the health system side in this space. So if you're interested in getting involved, please feel free to reach out. Just to kind of fly through the rest of these slides, this was the tripartite review committee that chose our platform technologies last summer. An incredible group, and you can see some people you recognize there, I'm sure. These are the technologies that were eventually chosen and the medallion architecture we're pursuing. One of note, Snowflake allows us to share data seamlessly without transferring data among the schools and NYP. So that provides us with a technical backbone, and then we'll have a common governance mechanism on top of that for how we transmit data. These are just eye charts. You can have the slides later if you want to see. This was just the full set of data systems that we are planning to integrate and the kind of rough timeline. I will tell you, this is like a cartoon, and we all know that there's many layers of complexity underpinning this, but we have a great team. It's headed by a director based at NYP, but she's working closely with data engineering leaders at both schools, and there's a lot of great momentum and energy there. These were just some of the cardiac AI projects that NYP helped fund and support. As you can see, there's a ton of great work here. It's very, very translational in nature. We know that we're a leader as a tri-institutional consortium in cardiology. I think radiology and other imaging domains are probably right up there with that. So we are excited to offer our support to this area. These are just some of the active data science working groups within the data shared service. And I didn't put this one up there yet, but it will be up there as well. So I will pause there, and I will take any questions. Thanks so much.
Any quick questions?
Yeah.
Since you have one hospital and two universities, is there any thought of like combining IRPs or like the fact that we need data use agreements between the two universities just to get research off the ground? Like, what are like some things that can make research across campuses more seamless?
Yeah, I think I'm a data scientist by training, so I like the technical part more than the people part, to be totally honest. I think having we talk about this a lot, in all seriousness. I know there's an effort right now at Cornell to consolidate like the Brooklyn and Queens IRBs under the Cornell umbrella. If you want to do tri-institutional research right now, you need collaborators at both schools. That kind of stuff gets complicated. But there is a track process that is working. I'm smiling because there's a faculty member here who would really love that question, who has been harping on me to build like a shared service IRB, like build a separate IRB that could be run through the shared service. I'm completely open to having those conversations. I just want us to solve one thing at a time. We already provide, that our EPIC data, you don't see it necessarily, but it's provided from the shared service already. And so this is another mechanism kind of on top of that that's similar with a similar governance structure just for other data sets.
Just so we don't get too far off time, maybe we will move on to our next speaker. But
Sounds great. Thank you so much.
thank you. That was a fascinating presentation that really did only take about one slide for the most of it. So thank you very much. Next up, we have Ruth Carlos, Vice Chair of Academic Affairs and Community Engagement in the Department of Radiology.
Thank you. I can't believe Despina is going to miss this because I'm going to say Ευχαριστώ Gia tin proklisi, which apparently means thank you for this invitation in Greek, which is the theme of today. This really is why we do what we do. The 5 R's, right patient, right test, right treatment, right time, and for the right price. And many of us in this room work in precision diagnostics and treatment. Our goal truly is personalized medicine, treatment at the end of one. But in order to understand all that goes into the experiences, the responses, and our ability to actually diagnose disease and monitor treatment, we need to understand in a person's life what actually constitutes risk. And the Robert Wood Johnson Foundation in 2014 note that even if we were perfect, our ability to contact patients appropriately is only about 20% of the time. The other 80%, their exposures really come from where they live, where they work, where they pray. Socioeconomic factors, physical environment, health behaviors, many of which are influenced by their zip code. So taking and integrating a lot of the information that we seek to package into our next AI program, multiple omics. This is one more omics, the sociome, or sociomics. This is where we start from the society, which influences the cell, which ultimately influences the outcomes. And we call this social genomics. And in order to understand the influence of social genomics on health parity, There's much epidemiological work that has been done in linking social context, institutions and access, social determinants of health, health-related social needs, and behavior to what we see as disparities in biological and clinical outcomes. What we are seeking to find is this part in the middle. How does one's individual genetic risk be influenced by upstream social contexts that may produce physiologic risk, as well as alterations in genomic and epigenomic regulation to produce the differences that we see clinically? And more importantly, how do we package all of that into an AI model that robustly represents the full experience of the individual patient? So when we think about social context and the effects on health, there have been some proposed mechanisms as to one social context affecting the stress response. For example, social adversity is associated with upregulation of stress hormones and secondary manifestations of stress disease. In the epidemiological circles, this has been referred to as allostatic load. There has also been a stress response related to genes involved in inflammation from social context effects. And these may lead to genomic and epigenomic changes that are both reversible and irreversible. There are also peritumoral and immune responses, upregulation of genes involved with epithelial mesenchymal transition, which may influence whether cancer is formed and whether cancer metastasizes. The tumors themselves manifest a variety of gene expressions which dictate how aggressive they are and how well they respond to therapy. Some of these solid tumor gene expressions have been linked to upstream social conditions, and this includes gene expression related to inflammation, wound healing, blood vessel growth, which governs potential metastases, and resistance to apoptosis or resistance to chemotherapy. As you can imagine, what we seek to model with our AI algorithms is a very complex experience that varies from the person you are to the person sitting to your right and to your left. And what that needs is data, robust, reproducible, equitable data. To that end, CIMBID and the Department of Radiology has partnered with the Columbia University Biobank, or CUB. CUB is a centralized resource that collects, processes, and stores biological specimens, biomarkers, and health data for use for CUMC researchers. The long-term goal, as Despina had articulated earlier, is to invite every single patient that we have in our Columbia NYP system to enroll using the IRB umbrella protocol. The consent allows us to access medical records, collect any excess tissue for banking, to draw urine and blood specimens, and to redraw and recontact for future research. The goal then is to provide a living model of how we collect the various types of data that are available and that can be used to inform AI algorithms and ultimately to inform care. Currently, institutional funding supports processing and storage of serum, plasma, and urine, and there are research partners who are encouraged to support recruitment and sample data collection costs. The radiology department is underwriting a CRC who helps recruit patients, and one of the innovations is that we have streamlined a recruitment process of patients who are attending a variety of imaging examinations in the Department of Radiology. Radiology has contributed approximately 1,000 patients in the last 6 to 8 months since we've started the collaboration. Underpinning CUB is a comprehensive informatics architecture. This combines sample data from the CUB biobank. It also incorporates genotype data, including CUB sequencing, It draws from central REDCap data sets that will include qualitative and survey data, as well as other data generated from shared outcomes, shared collections, in order to integrate this for access as a researcher. The collection is automated and scalable, and the goal for the radiology pilot later in the year is to recruit specifically through the patient portal linked to a patient imaging encounter. There are several innovations associated with this effort, which includes understanding the use of opportunistic encounters such as radiology for data collection, which is a potential framework for care delivery. It provides application of multimodal phased intensity recruitment for increased efficiency. And we are beginning a series of focus groups to understand return of results to patients, which represents an opportunity for dissemination and community engagement of some of our findings. Ultimately, though, the reason why we want these data is in order to address some of these questions and some of the potential questions that underpin social genomics, health parity, and predictive science are, can we predict epigenetic age from DNA methylation, and how can we use a functional age compared to chronological age to predict who is going to respond to therapy and who should be offered more personalized therapy? Can we use metabolomics, which has been shown to be susceptible to the environment, and look at body composition and treatment outcomes, as well as understanding whether adiposity represents a potential mediator for social exposure? And finally, we know that there is increased prevalence of coronary artery disease in high heat areas. Can we use these large data sets to be able to understand extreme weather events and how resilient our system is in being able to care for individuals that have increased need in the future? Once we actually answer those questions using AI algorithms yet to be built, then comes the tricky question of translation. This is the standard translation mechanism for data algorithms. This is how I prefer to view translation of data algorithms to the community. We have to start understanding what process in the clinical care episode are we actually trying to replace, and then understand how clinicians and AI actually interact. How do physicians view the data, view the output? How do patients view the output, and will it get the desired either behavioral effect or therapeutic effect? And then ultimately, how does the system respond to implementation of AI algorithms? For example, hiring practices, addressing de-skilling, et cetera. So ultimately, we end as we begin with the 5 R's: right patient, right test, right treatment at the right time and for the right price. And what we seek is AI parity, and underpinning AI parity ultimately relies on robust, reliable, representative data, hence our collaboration with CUB as we seek to move these data into CIMBID algorithms. Thank you.
Quick question? Great, thank you. Got one.
Hi.
Using that word. I guess, I know we're talking a lot about these large clinical data sets and things like that, but as epidemiologists, we have been working on these large cohorts for years and years, and, painstakingly collected all this data. Are there opportunities, for us to also leverage those kind of data to do some of the work that you're proposing? Or I feel like everybody's been framing more as like using our clinical data, but can you talk a little bit about that? Because you brought up like the social genomics and all of that, and that's the kind of data that we have collected rich data for, actually, the variables. Yeah.
So data are data, and I'm agnostic as to where the source is. There are definitely opportunities to integrate these large cohort data, or even state-level data. For example, the state of Maryland, which has an interoperability mandate, allowing them to collect EMR data across the state. So wherever the data come from, I believe that they will serve as a rich source as long as the data are actually there to inform algorithms. And one of the challenges that I find, especially in historical cohorts, is that the science has advanced, but we are still left with whatever they collected 20 years ago.
Thank you.
Thanks.
Fantastic. Next up, I'm going to welcome Shane Khalil from New England Higher Education and Academic Medical Center, the New England Higher Education and Academic Medical Center leader at AWS. Thanks.
Hi, good afternoon. Despina, thank you. Appreciate the opportunity to be here today. There was a scheduling error. We'd hoped to have our healthcare Director of AI join us today, but I'm going to try to do us justice on this message. I do have the opportunity to lead our Northeast Higher Education team and the engagement with the customer base supported by those account teams, including Weill Cornell and obviously Columbia Medical. Super excited to be here. I was asked to share a little bit about how we envision transforming and reimagining the partnership between AWS and Columbia University and really academia and industry in general. And from Dr. Gupta's opening remarks around the power of data to the obvious AI transformation that was on display today, we really kind of think that and reimagine it from an AI acceleration partnership, right? So what we have historically been is a cloud services provider, but what we look to be in the future and what was kind of coined by the director of AI innovation at Pitt last week at our DC Public Sector Summit was that we've become more of that AI acceleration partnership. So when we think about the scale necessary to impact the changes and all of the impressive science that you're bringing to bear, that's at its core and foundation. This will for sure be the least scientific presentation of the day, I promise you that. So, thank you. Where does it start? It starts with the dedicated team working more closely with your teams. This team brings deep expertise and personal connections to Columbia University. I'm joined by Angela Helfrich and Mark Bronneman, many of you which you know. Angela has been serving as CIMC's dedicated account manager for the past 4.5 years. She's a Columbia Business School graduate and actually welcomed her first child at CIMC. Mark spent 19 years at Weill Cornell, where he helped to lead their AWS practice before joining us 4 years ago. Together with my background supporting research across federal agencies and higher education, each team member represents AWS's investment in understanding and supporting the unique requirements and needs of the research community. So CIMC researchers have already achieved significant success with AWS. Prime example is GenBar team, who helped shape AWS Omics service while building the robust infrastructure for the next generation sequencing data. That work exemplifies how our partnership in advancing scientific discoveries and cutting edge capabilities is a two-way street. Your requirements are influencing the way that we're developing our services, specifically within AI and across healthcare AI. Our vision is to accelerate your medical breakthroughs by providing immediate access to that world-class cloud infrastructure and specialized tools from early career scientists to established investigators, giving access to that scalable computing power in a HIPAA-compliant storage, with HIPAA-compliant storage, advanced AI and ML capabilities, and genomics workflows, all supported by dedicated technical teams and investments. You know, on the heels of a $20 billion investment announcement last week with COPA, the connections that we're drawing between the medical community, the AI development community, and all across higher ed within that state is just one great example of what we can be doing here within New York and what we will continue to be doing. We will support your work through the research lifecycle. We long have. Starting with strengthening grant proposals, proof of concept testing, mapping industry and sponsored research, so finding that funding and aligning it to your important research. And as funding is secured, researchers can quickly develop and deploy their environments, accessing that high-performance computing need and the data infrastructure management tools that are necessary. And that accelerates everything from enabling complex analysis from genomic sequencing to image modeling, while maintaining strict security compliance standards essential for CIMC and other research centers. As discoveries emerge, researchers can seamlessly share their findings through secure global networks, fostering international collaboration while maintaining that data governance that's necessary. This is the infrastructure that Despina referred to earlier, at scale. Success breeds success. As researchers projects demonstrate impact, they attract additional funding and partnerships, feeding back to the flywheel. Each revolution strengthening Columbia's research capabilities from initial concept to global impact, while reducing administrative overhead and maximizing the research outcomes. In strong partnership with CIMC, our startup team can connect these breakthrough discoveries to incubation pathways that they need, helping them to efficiently transition to market-ready solutions, generating resources to fuel the next wave of innovation. I think back to Garud's discussion about DSI and related to the education theme and partnership. We believe there's a great need to continue to connect those researchers and technologists alike to the skills necessary to think, I'm thinking industry mentorships, scholars programs, training. This partnership is about amplifying Columbia's research excellence, supporting your mission to advance knowledge and learning at the highest level. Together, we'll help Columbia's researchers push the boundaries of discovery and innovation. To support Columbia's research excellence through these challenging times and unprecedented times, we're offering significant investments through AWS research credits. Programs that are in place today, as well as new programs like a new junior faculty research package. Unlocking investments that demonstrate our commitment to ensuring Columbia's vital research continues unimpeded, allowing researchers to focus on their critical work. And we will continue to align these programs to CIMBID and DSI and the likes. So in closing, this partnership represents more than technology collaboration. It embodies a shared vision of advancing medical sciences. Together, we can create a model of industry and academia partnership that sets the new standards accelerating scientific discovery. Thank you for the opportunity to be a part of today, and congratulations to Despina and team.
Certainly these are challenging times, but it is wonderful to have this ongoing, rich, really robust, supportive partnership. So thank you very much for being here today. Finally, I'd like to introduce Greg Goldmacher, Clinical Research and Head of Clinical Imaging and Pathology at Merck.
All right, so I'm the last guy. I'm the last guy between you and a coffee break. I'll try to be quick, and I will be. Okay, so I got to industry from academia about 15 years ago, and I thought, okay, so in clinical trials, we do the most advanced complex imaging, right, with all of the measurements. No. In a phase 3 trial, I want to have imaging that can be done in Chile and in Ukraine and in, basically all over the world. So we do not do advanced imaging, certainly not advanced imaging analysis, okay? What we do is really simple imaging in late phase trials. Oh, by the way, sorry. So I'm Greg Goldmacher. I'm a radiologist by training. I lead this group called Clinical Imaging and Pathology at Merck. So that all of the endpoints, meaning the outcome measurements in clinical trials, that's what I oversee based on scans and based on biopsies. What I want to tell you about is basically what it is in industry that we need beyond these very simplistic things that we're doing. And by the way, this is what I asked GPT to produce an image of AI reading an X-ray. There we go. That's an AI radiologist. Okay, so look, pharma basically has two big parts. I mean, there's actually, well, three. So research and R&D, developing drugs. commercial, selling drugs, and manufacturing, the actual making of the drugs, okay? So every big pharma has those three components kind of joined together somehow. I don't know anything about the manufacturing side, so I'm going to tell you about some of the needs that can be met by AI-supported measurements, mostly in development, because that's what I do. I have a little bit of touch points with the commercial side. So first of all, here's a big problem that pharma has, and that is that the simple measurements that are made in late phase trials, the things you need for the FDA submission, okay? Like, so in oncology, objective response rate or progression-free survival by RECIST, these kind of endpoints in oncology, they correlate well with what we really, really care about, which is survival at large N. And at small n, they don't. And there is a long history in cancer drug development of drugs that looked good based on shrinking a sample of tumors in their aggregate diameter in phase 2 and then failed to improve survival in phase 3. That's a disaster. First of all, for all the patients, right? But also even for the pharmas, because that's a few $100 million down the drain, okay? So how do we need basically better measurements of efficacy with small cohorts. So if I can, because we make big decisions based on what is unreliable data, essentially. We make decisions in phase 1B and phase 2A where you're only spending like tens of millions before you commit to that program in the hundreds of millions, the late phase program. So that's one major need. And what can you do with that? So with AI, radiomic signatures that tell you something about tumor inflammation or vascularity or things that the human eye can't pick up, but models can, and can be used as a biomarker. That's kind of the second category. Or if you look at changes in that to give you some early readout on efficacy to get closer to the biology. Or things like tumor growth kinetic analysis, right? Where even with things like by exponential kinetic models, you get much closer to survival. The problem is you want to measure every tumor in 3D because any sampling-based strategy like RECIST means when you pick out a few tumors and you measure those, the assumption is the outcome of interest, survival, is driven by the average tumor. And that's not true, right? It's not the average tumor that kills the patient. It's the most aggressive, most treatment-resistant one that's growing in spite of treatment. Okay? If you don't happen to pick that one, you're blind to it. So we need something that'll, but radiologist time is expensive, right? So finding everything, and segmentation is a solved problem largely, but basically finding every tumor on every time point. That is something that is doable with AI. I mean, it's in the near term, even in the near future. So that's, those kinds of measurements will enable us to make better decisions in the early pipeline for pharmacodynamics, for early clinical efficacy, for picking dose, which is a huge deal in pharma development, choosing the right dose. And then in large-scale trials, there can be use of AI for things that are more basically more of a pure efficiency play, making measurements at scale that are expensive. So right now on a phase 3 trial, I spend about $5 million on just the imaging review, okay? So if we could automate a significant portion of that, that would be a nice savings. I mean, it's still a small component of the overall trial cost, but every bit counts. And then in the commercial space, all of the diagnostic stuff, the making, opportunistic detection of early stage lung cancers or of pick your cancer type or pick your disease type in general. Early detection does give you an opportunity to intervene. I will say that for pharma, obviously, pharma decision makers are a little bit blinkered in the sense that, they care about early detection when there's an opportunity to treat. And so, if something is detected so early that there's no opportunity to treat, pharma isn't as interested. But there's certainly an opportunity also for, in that early diagnosis, to also make additional predictions of future behavior. So if you have an early stage tumor that's detected, and it's just going to be removed surgically and that's it, okay. But if there is a risk, if there's a way to look at the scan and with some set of radiomic features stratify the risk of recurrence so that you can identify, hey, these patients are high risk. You do the surgery, that's probably not enough. You want to treat with adjuvant therapy. Okay? So those are the kinds of applications so that pharma has a need for sort of advanced image-based measurements, advanced analytics, to serve as novel endpoints, to serve as novel biomarkers, and to serve to some extent as detection tools. So what we gather is a large amount of data. Some of it is advanced data, some of it is not. But digging through all of that, so bringing it together, harmonizing it. The nice thing is we already typically pay for the scans to be collected, cleaned, stored. So we have large data banks of data that's sequential, curated scans at multiple time points in a clinical trial. So that's kind of an asset that pharma has. Pharma also has internal resources for research, people who do this kind of stuff. However, we also have a hard time doing multimodality work internally because things tend to be quite siloed. And so, in an academic center where you're able to bring together data across multiple modalities, that's actually an area where you may have better opportunities to do research and develop models than we do. Although, one way that I see this working is you guys develop models and then we take them internally and test them on our data, and then we sort of collaborate on how to implement them. So that's all I have. I'll take questions. And just, once again, Despina, thank you for bringing me here. This is
This was really interesting. So I know that some things they have to take a certain amount of time because the FDA, and you want to see if certain side effects occur. So with all of that in mind, how much faster do you think it could be, from the start of R&D to the drug actually being made available to consumers? Do you think we could cut in half by 50%, or just it's up to the FDA for the most part?
That's a really hard question. A lot of safety is determined initially in early phase development, and then you continue to monitor the incidence of safety in late development. But in terms of cutting development time, I think maybe 25 to 30% is my guess, because of that's a guess. Don't hold me to that. The reason is all of the late phase development that takes quite a bit of time. The limitation there is not analysis; it's recruitment. So you got to recruit a certain number of subjects. If AI enables us to develop better surrogate endpoints, ones that are so close to the biology that they're truly, eventually with FDA acceptance, accepted as a surrogate endpoint that's approvable, then potentially you could cut the recruitment number down. But right now, the main the main savings I see is that early to mid phase where you have to make decisions, and because the decisions are very high stakes, it can take a while to accumulate the level of data that gives people confidence. So if we have better measurements, this is a sort of an easy mantra: better measurements, better decisions. And better measurements, better decisions can mean decisions of the same quality but with a smaller data set.